Hi, Dr. Brian Koffman here in San Diego for ASH 2023 for the CLL Society.
And I'm Dr. Adam Kittai from The Ohio State University, where I specialize in the treatment of patients with CLL and Richter's transformation.
Well, the CLL community in the last few weeks, in the last week, has gotten really good news about an approval of a new drug, pirtobrutinib, which works when the CLL cells have become resistant to the covalently binding BTK inhibitors. Could you talk us through a little bit about covalent, non-covalent?
Yeah.
And if we can get into, there's a new drug that's being presented here, LP16H, which I understand binds both covalently and non-covalently. So help us understand this clonal evolution and all these mutations. It gets pretty complicated. There's a lot of letters and numbers in there.
Yeah. So we've come a long way with targeting the kinase, the Bruton tyrosine kinase inhibitor. So we started with ibrutinib, which was our first generation drug. Then we got acalabrutinib and zanubrutinib, which was our second generation version of ibrutinib, that bound the same way to BTK that ibrutinib did, but appeared to be safer and maybe more effective. Then we have our third generation, which binds slightly differently. That's the the covalent way. And now we have this new drug that's our fourth generation, which is LP16H that we'll talk about. So when patients get either ibrutinib, acalabrutinib, or zanubrutinib, drugs bind covalently to the BTK. And the CLL cells rely on BTK to signal and survive. So this is a key kinase for the cancerous cells.
Kinase is just an enzyme.
Yeah, exactly. So it causes signaling through the cell that causes the cell to divide faster and live longer, which are the two defining features of cancer.
I say it like there's an antenna on the surface of the cell and sending a message saying you're important, stay alive, and have lots of babies.
Yes,
yeah,
exactly. That's a good way of putting it. And ultimately, our cancer cells rely on over-signaling through that antenna.
Right, they're addicted to it.
Exactly. And so these three drugs were basically putting a brake on that antenna, blocking the signal from the outside of the cell into the inside cell.
Right.
And they all bound covalently to the BTK.
Exchanging a hydrogen ion.
Exactly. Good biochemistry. And what happens, though, is that across the cancer world, when we have drugs that bind specifically to any kinase, what happens is because the cancer cells continue to divide, eventually a mistake happens where a mutation occurs where the drug is binding to the BTK, causing it to no longer bind as well. And that mutation that you may have heard about is something called the C481S mutation. And this is the primary resistance mutation that leads our patients to start to progress or get worse on ibrutinib, acalabrutinib, and zanubrutinib. So in comes along pirtobrutinib, which is the drug that was just approved, which doesn't bind covalently, but binds non-covalently, and should overcome resistance to patients who've developed that C481S mutation. And so just like these resistance mutations occurred with the covalent BTK inhibitors, we're now seeing resistance mutations that occur in the non-covalent BTK inhibitors, specifically a mutation called T474I. So now that pirtobrutinib is approved for CLL, there's going to be a group of patients who've now received pirtobrutinib who've developed this mutation and are now progressing on pirtobrutinib. So it's important to think about, well, what's next after pirtobrutinib, and that's where LP168 may be able to fill the spot. And so LP168 is a unique BTK inhibitor that binds both covalently and non-covalently and can kind of switch on and off.
So would it work in the presence of both of those mutations?
Yeah, so the abstract that we're talking about today is the preclinical work that was developed in the lab of my mentor, Dr. Woyak, and one of her postdocs, Britten Gordon. And so Mr. Gordon is presenting this work here at ASH as an oral, and what he looked at was...
And oral presentations are the ones that are thought to be the most important, of the most interest, the most advancing the science.
Exactly, as
and
opposed to a
very
poster.
hard to get.
Yes,
yeah.
as opposed to a poster.
Right.
And what he looked at was CLL cells in the lab that had both the C481S mutation and the T474I mutation, and looked to see whether LP168 was able to inhibit those cells. And what he showed was, in fact, it was able to inhibit those cells. And so based off of this work, we now have a clinical trial that's also being presented by Dr. Woyak here at ASH that looks at the use of LP168 in actual patients as opposed to in the lab and with mice.
Yeah, and we'll be speaking to Dr. Woyak on that paper to see what those results are in patients and after it's presented here at ASH.
Yeah. And one of the things to know here is, one of the interesting things here is that not only does LP168 bind both covalently and non-covalently, it's super specific for BTK, meaning that we should hopefully not see as many of those off-target effects. And what off-target effects are is when we have a drug, it always not only inhibits BTK, but can inhibit other kinases, which might lead to side effects. And so that's why the second generation BTK inhibitors, acalabrutinib and zanubrutinib, appear to be safer than ibrutinib because there's less off-target effects. Here, LP168 is very specific for BTK, hopefully leading to much less off-target effects.
The cardiac and the bleeding issues and things like
exactly,
that.
And the hypertension, everything.
Are there clinical trials of LP168 open right now?
Yeah, so the LP168 trial is currently open at The Ohio State University, the University of Cincinnati, and Utah, I believe. It might be open it's also open, I think, at Duke as well, but I'm not 100% sure on that.
Any final thoughts on this evolution of the resistance to the BTK inhibitors?
Yeah, I think that the question that comes up is whether or not resistance will actually be deployed in non-academic settings. And I think ultimately the question comes down about sequencing, like how should we be giving these drugs in order? And to me, if we're sequencing all of these drugs that hit the same kinase, I think it's a win, right? If we continue someone on these drugs that are really safe, they're doing well on them, and they can go from acalabrutinib, zanubrutinib, to pirtobrutinib, to LP168, to a degrader, which also targets BTK inhibitor, or some other future BTK inhibitor gets developed, before they get on to targeting a different target, and they're tolerating all these drugs really well, I think that's a win for our patients.
I agree. I think the more options we have and staying with one pathway, I think really makes sense. I think this is an exciting area of research, and it does involve a little digging, and it's really great to see this kind of preclinical research. That's why I think this stuff is so important for patients to understand, not just the results on how many patients get a response and what the duration of the response, but what's the science underneath it to understand it. Dr. Kittai, thanks so much for all that you're doing. Thank you.
Thanks for having me.